in the lab. Yeah. So Patrick is a visiting scientist in the lab of Ada Klipp and Roon Linding at the Humboldt-University of Berlin. And I would give you the stage.
Thank you so much. Quickly share. So, hello guys, my name is Patrick Brunner. Thank you for the kind introduction. I'm a guest scientist in Rune Linding and Edda's lab. What I'm gonna present to you today is me looking at the spatial simulation of cell signaling using steps, or in a more biological sense, we're investigating the influence of cell shape on the EGF-ERG pathway dynamics. This whole project has started with Anna Truppenbrot here in the second, when she did this work in the lab or in our lab. And I kind of took it over as my passion project. I did my PhD in Edda's lab, and at the end of my PhD, I stumbled upon this project, found it very interesting, and then now kind of continued. Besides my other job, I've been doing this project with it because I just think it's very interesting, and I kept the interest up on it. So I'm very glad I can present this here. It's a great opportunity. So what we're looking at is the MAP or ERG pathway. I mentioned ERG before. There was a renaming some years ago, so it's now called the MAPK pathway, but it's the same thing. And we're interested in this pathway and the disease it holds. So it's a very evolutionary conserved family of signaling pathways. It's relevant in cancer research as it's dysregulated in many human cancer types. And it's about cell proliferation. It regulates cell proliferation, and as you can imagine, cell proliferation is very relevant in cancer growth as it can facilitate cancer. If we look at the pathway, or at least the version of the pathway we are interested in, it's a protein kinase cascade. And in our version, it starts with the EGF, the epidermal growth factor ligand binding to the receptor, here the HF receptor, and then through a couple of proteins down into the ERG, PP, double phosphorylated ERG, which arrives in the nucleus of the cell, and then in the end activates transcription factors. What's important about this pathway regarding the disease is this RAS here. Acts like a switch. It is in this process, it is turned on, and if the signaling has finalized, it will be turned off again. But under disease, it can remain activated. And as it regulates cell proliferation, it can lead to increased cell growth, which is, of course, not that good in cancer. And that's why it's interesting for us in the cancer research. So what we're interested in now is the role of geometry in this whole path. So signal transduction, I've just shown you, is not an isolated process by itself. There are many confounding factors, but what we're interested in is the geometry. So what we're looking at is what happens with this pathway I just shown you under different geometry conditions. So different shapes of the cell, so to say. If we look at, we have three exemplary cell shapes here. We would have a difference in local membrane curvature. For example, here on the left, the membrane is very curved compared to the top here. We have a different surface to volume ratio between the sphere here and this ellipsoid. And we can have wildly different cell shapes. Just one example would be a cell with a protrusion right here, forming this shmoo. This could be one of the many cell shapes that would be relevant in the context of geometry. So what we want to do, or why is this interesting? Because cell shape does change, of course. I think most of you know that cell shape changes during, for example, cell migration, which is again essential in metastatic growth in cancer. And it would be key for us to understand how this geometry affects both the healthy and the diseased cell behavior. So the issue is with all of this, it sounds great, but how can we actually investigate this influence of cell shape? Experimentally, this would be very difficult to do. There's so many confounding factors. If you just think about it, signaling can be influenced by cytoskeleton, active transport, lipids, rafts, different concentrations of receptors on the cell surface. There are many confounding factors, and experimentally, this would be very hard to control if you want to just look at the role of geometry. And because of that, and because we are a modeling group, we chose to do a model. And we use model as a tool to investigate just the role of the geometry. The first intuition, if you want to do a model about something like this, would be to do it as simple as possible with an ODE. ODEs are ordinary differential equations, but what you have to do with these, you would assume a well-mixed system, which is a system that assumes that, for example, the EGF concentration would be homogeneous all over the system. And also within the cell, everything would be homogeneous. But this doesn't make any sense if you want to look at the geometry, at spatial information. So we couldn't use this well-mixed system. We had to go spatial. And another factor is that these EGF numbers in the beginning, they're quite low. We have very low initial EGF numbers, so in the hundreds. So we also need stochastic signaling, stochastic modeling. So we chose spatial stochastic modeling to capture the stochasticity of the first binding of the EGF to the EGF receptor. And here on the right, I've shown you how that would look. As a cartoon, we have the cell here. We have different compartments like the cytosol, the nucleus. And what we do with this whole volume of the cell, we then create subcompartments called voxels. So these little volumes here. This is a 2D slice. These would be volumes, so in 3D. And within these volumes, there can be different reactants, different species. These would be the species of our signaling cascade. What they can do, they can be in these compartments, they can move from compartment to compartment, essentially diffusion. So we would have diffusion, and what they can do here on the right, they can react. So they can, for example, these two species can react then to form a third species. So this allows us to model diffusion and reactions, and we do this via STEPS. STEPS is a package called Stochastic Engine for Pathway Simulation. It's just a toolkit we use to model the reactions and the spatial stochastic reactions we're interested in. Yeah. So this is the full-on ERG pathway model. This is not the simplified ERG pathway. This would be the full pathway, and it's very complex. And there's around over 60 species in there. And to start off with a pathway like this would be very challenging and very hard, as spatial modeling is still very computationally expensive. So we chose, as a first proof of principle, we chose a simplified and shortened version of this pathway model. So here I've shown you on the right the simplified model. We have still EGF on the extracellular space binding to the EGF receptor, that's EGFR, which then dimerization, then there's phosphorylation down here, binding with GAP, ERG, and then down into the nucleus. So we went from over 60 of around 60 species to just 10 species, which heavily reduces computational cost. So in my PhD project, I also did spatial modeling, and I know that large spatial models are very computationally expensive. So it's honestly necessary to start with a small model if you wanna get a feeling, as we say, for the model. And this small model can also inform us about potential influencing factors, which we maybe did not account for when we planned this whole project. So that's why we had to start with a proof of principle shortened version, and we did that. So we also chose 3 simplified, again, cell shapes. I showed you these 3 before. We have the sphere, the ellipsoid, and then just the sphere with the protrusion. These 3 all have the same cell volume, so around 1,600 square microns, but they only differ in shape and surface area. So we wanted to control for the cell volume to also not be a confounding factor, just have the difference in surface area and shape. And these, again, represent changes during adhesion or migration of the cell. So if you look at the one singular cell, what they have in different compartments, we have the extracellular space, that's where the EGF, we have the cell membrane, cytoplasm, and nucleus. I've shown you this before. And what we did, we kept the concentrations of the initial species. For example, here, these are the species in the simplified model. We kept the initial concentrations constant. So not the amount, but the concentrations. So this ensures we have the differences we find between these geometries are attributable to geometry alone. This is very important because that's what we're interested in. So if we look at the results, let's first have a look at the first result. Let's have a look at the sphere versus the ellipsoid. So sphere in blue, ellipsoid in orange. For example, EGF binding to its receptor over time. We see that the ellipsoid has a stronger or has more species here, over 15,000, compared to the sphere. And this continues on for the first 5 species we have here. So this is already interesting. Just through the change in geometry, we see a change in the signaling. And you could say that the signal propagates faster in the ellipsoid. So why is that? Probably because the ellipsoid has a larger cell surface area than the sphere. And so this is the only difference between these two. As we kept the concentrations constant, larger cell surface area means we have more initial EGF receptor as concentrations are constant. So more receptor means more EGF can bind to the receptor. We have more species, and then this results in a stronger signaling. But if you, as you might have seen, looking at the ERG-P in the nucleus, the final step, ERG-P diffusing into the nucleus, the ellipsoid is slower. So this is already interesting. The first one we kind of expected. This one was already interesting. This could be due to the longer distance the signal has to travel in the ellipsoid. So ellipsoid, we have a larger distance between the membrane and the nucleus at the outermost points. In the sphere, the distance is homogeneous. But it was still interesting, so we wanted to investigate that further. So what we did, we looked at local stimulus. So what we did, we have the ellipsoid here on the right. We looked at signaling from the top. And if you signal from the top, you see this is the nucleus, there's the membrane. The distance between the membrane and the nucleus would be minimal. And on the left, we would see the distance between the membrane here and the nucleus, if we go straight, would be at a maximum. And this allows us to see, hey, what's the influence of local signaling? And if we see overall, yes, if we look at the final score, we see that ellipsoid left, blue, is much lower than ellipsoid on top. It's very interesting. So this shows us, yes, the localized signaling is here much faster. In the blue, it has barely even started as the signal needs to travel all this way. But on top, we're already there. We're already signaling. So that was quite interesting and showed us, yes, okay, even with the small model, we can already see not only an influence of the geometry, but also an influence of the signaling location. And this is approximately where I came into the project. So what I did is so far try to build a robust and scalable simulation framework or pipeline. So what we want to do is, of course, use the whole model, the large model here, move from 10 to 58 species. But this is a significant technical hurdle, and I'm still working on that, to be honest. So what we want from the new pipeline, we want it to be able to implement a complete pathway, of course. We want geometric flexibility. That's a tough one. Before, with these simplified shapes, we could create them ourselves and put them into the pipeline. But we want to move beyond that and to include real geometries which are derived from microscopy data. And while doing all of that, we need to minimize the computational costs, utilize parallel computing. It's quite important, as running the full model can take up to days, just one run. So that is quite important to keep the computational performance at its best. And we have done it. I can show you a quick preview. I have been able just recently to do the full model. I don't want to go into detail because I'm not sure if it's correct yet. We still have to do some investigation here, but we are at this point where you can do the full model, and we have acquired realistic geometries, so real cells, courtesy of Chris Baker lab, where we have actual cell types, different cell types under different drug conditions, even different cancer drugs, where we can see, okay, how does the drug change the cell shape? And then we hope to, if we implement our model, we can see how does the drug change the cell shape, change the geometry, and then the signaling downstream. So that's actually what we're looking forward to doing. So this is what I'm interested in, at least. Of course, first we also have to do a parameter scan over the small model results. But what keeps me going is trying to implement these 3D cell shapes from the microscopy images of cancer cells. which then would connect our model, which just we have right now, to actual experimental data. And I think that's the most important thing we still have to do. We wanted to connect the theory with the experiments. And then we can explore more realistic signaling scenarios like the localized EGF stimulation I've already shown you. Then there's uneven receptor distributions on the membranes. There's more inhomogeneities you can introduce into the system to see what happens in the system. And then in the end, we want to apply the framework to investigate the migration of the cancer cells because it's about metastatic growth, for example, in breast cancer. But I think there are many applications. Yeah, that's the state of the project so far. Thank you for letting me present this passion project of mine. It's very, very cool to do that. And I'm now open for questions. Thank you.